again. I thank you to continue with you now. Now we are going to discuss about glucose as a structure component. Glucose as a structure component, you see, we are finding the word structure and the word component. When saying the structure, we need to mean how it is made. When it's talking about the components, what are the elements found in it? You see how its structure is. Now, as we discussed above, glucose is the main source of energy in all living cells. But this being regarded as the universal fuel, glucose also acts as the source of carbon for all the carbon-containing compounds of the body. You see, as the importance, we are saying the glucose seems as the universal fuel because it is the one producing the energy for all living organisms. And now it is said to be the source of carbon in all carbon-containing compounds. Okay? Here we are having example. We are having the ribose. The ribose, we remember, ribose here it is a pentose monosaccharide used in the synthesis of nucleotides and nucleic acid. Is synthesized from glucose through pentose phosphate pathway. Here now you are seeing how glucose is important. Other important compounds including glycoproteins. The glycoproteins, it is a protein with oligosaccharide chain or glycan chain. It is covalently attached to their polypeptide side chains. Okay, in glycoproteins we observe how glucose is important. Now we are seeing proteoglycan. Proteoglycan, it is a special class of glycoprotein which contain about 95% polysaccharides or glycosaminoglycan, and it has 5% protein and various other polysaccharides like cellulose, chitinase, and glycogen. All those compounds said, most of them made of glucose. Now you see glycoprotein are the integral membrane protein which plays an important role in the cell-to-cell interaction, where the cell can sharing with another one through those membranes. Now we see, for example here, an antigen-antibody interaction in the blood. In the blood type compatibility. It is mediated by glycoprotein, which determines the blood type of the individual. And you know, the proteoglycan on the other hand, are the major components of extracellular matrix. That's understandable, I think. Let's continue also. We see glycogen now. We are talking about glycogen. What's a glycogen? Glycogen is an homopolymer of glucose in which each molecule is linked to each other by 1-4 glucosidic bond and branching through 1-6 linkage. Understandable? We know first glycogen seems as the combination. It is a great chain of glucose. Glycogen is like a mode of storing glucose. When glucose comes in excess or in a great amount in the body, get stored in the get converted in the glycogen, where it is going to be stored in the body. It is synthesized in the liver cells. The process of biosynthesis of glycogen from glucose with the help of the enzyme glycogen synthase. With the help of the enzyme glycogen synthase is known as glycogenolysis. Here the process called to be glycogenolysis. The glycogenolysis, it is the process by which glucose get converted into glycogen with the help of an enzyme called glycogen synthase. In response to decrease in blood glucose concentration, glycogen breaks down to glucose 1,4-phosphate and glucose in the liver, and in the liver. In response to decrease in blood glucose concentration, glucogen breaks down to glucose 1-phosphate and glucose in the liver and muscles. This process is known as glycogenolysis. Glycogenolysis here, it is a process by which glycogen breaks down to glucose. This is done within the presence of the enzyme called the glycogen phosphorylase, which helps the breakdown of glycogen into glucose. We know the glucose are the smaller molecules of glycogens, and these enzymes removes glucose residues sequentially from the glycogen to yield glucose 6-phosphate. Other enzymes, glycan transferase and the glucosidase help in the breaking the branch forming alpha 1,6 glucosidic bonds of the glycogen molecules. The overall final product of the glycogenolysis is glucose 6-phosphate, which is converted back to glucose by glucose phosphatase and release into the bloodstream. You see here, what I need you to know, we are having how the glucose get converted into glycogen by glucogenesis, and we are seeing how glycogen get converted to glucose by glycogenolysis with the help of an enzyme called glycogen phosphorylase, glycan transferase, and glucosidase. All those enzymes helps the process of conversion. And then we are seeing the other one, which said to be glucose phosphatase, which releasing glucose into the bloodstream. So now let us stopping also to help you by generating the questions and then asking as I'm generating the answers for you, where not understood, where not taken well. I'm there for you. Thank you.